All right, so we've now worked through the basic underlying ways that we get epistatic ratios. Certainly how genes interact. We talked about the fact that to have epistasis, you need two loci, a couple of alleles at least at each locus, and then they interact some way to affect a single trait. What I thought I'd do in this exercise and in part 5 is to work some epistatic problems. Now here what we're going to do is we're going to strip away all the words, at least for the first part of this, and not worry about that. We're just going to try to say, what's the underlying principles, the ideas that you need to work an epistatic problem? We'll finish up with a slight word problem, but then on part 5, I'll really go into more complex problems with real paragraphs where you have to pick out information. Okay, so just to think about what we need to do here. Okay, one of the things that I recommend for people is that we can use this 9-3-3-1 ratio as a building block for all sorts of ratios, okay, epistatic ratios. And if we know that, and we can kind of write this little thing down here, 9-3-3-1, big A blank, big B blank, 3, big A blank, whatever, little a, little b goes with 1. Write that down. We can actually make up a number of different types of ratios, so we don't have to really memorize them. We can do it. Now, you won't get all the epistatics out of this, okay, by any means, but you can get one of the big ones that you tend to see in a lot of the problems, particularly in textbooks and places. So think about this. If we want to build a 12 3-to-1, okay, we want to make some kind of single dominant epistatic. Well, we can do that by saying, okay, I need 12 parts, all right? Well, one part can come from right here, little a, little b, little b. One of the 3's can be held out, okay? And then how do I get a 12? Well, a 9 plus 3 will give me 12. Then you have to say, well, what's in common here? Okay? Well, what's in common here are these big A blanks, right? It doesn't matter what the B locus is doing. I've got big A that does something, turns it on or turns it off. Okay. Now I could have used this 3 down here. In that case, big B would have been in common. Doesn't make a difference as long as you pick one and stay with it. Okay. So I want to make a 12-3-1. I can simply do that. My 3 comes from there. My 1 comes from there. Maybe I want to make another ratio, a 9-3-4. Well, there's a 9 right there. I don't have to change anything there. I can grab one of my 3's. It doesn't matter which one. I happen to grab the one with the big A and 2 little b's. That gives me another phenotype, right? That's a genotype that's going to give a phenotype. And now I need a 4. Well, how do I get a 4? Well, 3 plus 1 will give me 4. What's in common between these two? It's the fact that they both have little a's. So I can just use the little a's and say, okay, and I could have picked again little b's, doesn't matter, but that will give me a 4. So when I do an epistatic problem, and I think this looks like a traditional one, I'll often just write down this 9-3-3-4 little limerick right here. And then I can kind of build it up. I can make 15-to-1s. I can make things like 9-to-7s. I can make all sorts of different ratios by just combining them up and taking a look and not having to memorize so much. Okay. All right, so let's try a problem. Let's take single recessive epistasis. We already talked about this, right? We talked about this being the mouse, the Goody gray, black, or albino. In this case, we've got one locus here. We've just symbolized it as A. And if you have a dominant A, a big A, then that allows color to be turned on. If you have the recessive condition, that's why it's called single recessive. If you're in the recessive condition of that locus, it just turns off color. It doesn't matter what the other alleles locus is trying to do. But if you have a big A, then you look over here and say, okay, at the B locus, what have I got? Well, if I got a big B, I'm a Goody, the gray. If I'm 2 little b's, I'm black. Well, but look here, I don't care. I'm a bino anyway, all right? So think about where this ratio comes from. This 9-3-4 comes from crossing a dihybrid heterozygote, big A, big B, little b, right? Times big A, big B, little b. That'll give you 16 parts. 9 plus 3 plus 4 is 16 parts. And that's where we get these ratios. That's what we did all throughout those first 3 lectures, all right? Now, let's think about doing something else. Let's take a cross that doesn't involve 2 dihybrid heads, right? Here we have a dihybrid head over here, but on this side we've done something different. We now have a homozygous little a for the first part and then heterozygous over here. All right, well, the first thing we want to do is figure out what these mice look like, right? Well, this one has a big A and a big B. You just look at our chart up here, that says, well, that one ought to be an agouti mouse. And then this one has 2 little A's. Well, it doesn't matter what the B's are once you have that, it should be an albino mouse. And sure enough, we have a Goody and an albino. So that's basically what we're crossing, okay? Now, notice we're not crossing 2 heterozygotes, two agoutis, we're crossing something else. We're not going to expect to get 16 parts. We're going to get something else. But guess what? Remember old Mendel right here? Hasn't failed us. Doesn't fail you when you get the epistasis, okay? Just look at the crosses and work with it. Now what you've got are two loci, an A locus and a B locus, but you can use the two basic Mendelian principles. You can use the principle of assortment to allow you to take and separate those out and work with them independently, those two loci. And then you can take one of the loci and work with those independently to give the segregation ratios, right? And that's all I did, okay? I just said, okay, what I'm really crossing is big A little a times little a. That's a 1-to-1 genotypic expectation. At the B locus, I'm crossing two hets. I get a 1-to-2-to-1. Now I'm just going to use, you could use Punnett square, whatever you want. I'm going to put those together and just put these two together, that together, and carry down all the numbers that I need. Okay, so I do something like that, write all that down. I'm going to get 6 parts. How do I know that? I've got 2 parts here, 1, 2. I've got 1, 2, 3 big parts here. Don't worry about what the numbers are, just 1, 2, 3, and 2. 2 to 3, 6. So I expect 6 different genotypes. Okay, and that won't be how many phenotypes I get, that's just how many genotypes. And sure enough, there's all 6 of them. Okay, all right. Now, what do I want to do? I want to pull down the numbers. This times that's 1, this times 2 is 2, etc. We've got all our numbers pulled down nicely. Okay, now what do they look like? All you got to do now is look up at the chart. You got a big A and a big B, that's a agouti. A big A and a big B, doesn't matter what the other allele is, that's another agouti. A big A, little a, but 2 little b's, turns out to be a black. And then all these guys down here have little a's, so they're going to be albinos, right? So we just sit there and put all that together. We take our genotypic ratio and we say, okay, how's that going to look as a phenotypic ratio? Well, we got 3 agoutis, right? Because these are all agoutis. We've just got 1 black because that's the only black genotype we could get. And then all these guys down here get piled together and we see them as albinos. So what we end up with is a 3 to 1 to 4 or 1 to 3 to 4 or 4 3 1, whatever you want, order you want to put it in. That's the final ratio. Now, what kind of ratio is it? Think about it. You can answer. What ratio is this 3 to 1 to 4? Okay. Well, it's phenotypic, right? It's the way things look. And it involves more than one locus, so therefore it is an assortment ratio. Our final phenotypic assortment ratio for this cross, where we cross this type of agouti times that type of albino, is a 3 to 1 to 4 or 1 to 3 to 4 or whatever. Okay. All right, let's try another one. Let's take a single dominant epistasis. Well, this goes back to those squash that we were using, right? In this case, we've got the reverse in our A locus. In this one, if you get a dominant A, it turns off color. So we see them as white squash. If you get the recessive condition, 2 little a's, then it allows color. We look over the B locus to see what the color will be. And it can be either yellow or green. Okay. Again, 12 plus 3 plus 1, 16 parts. That ratio is produced by a dihybrid het times dihybrid het. Okay. But let's try something else. Let's take it and say, okay, for the first individual we're going to cross, it's big A, little b, little b. All right, well, what's that going to look like? Well, it says up here, if you've got a big A, it doesn't matter what the rest of it is doing, you're going to be white. So this side's going to be white. And this got 2 little a's, so now it's going to have color. What color is it going to be? Whatever color's over here is being allowed. That's yellow. So in our case, we're crossing a white times a yellow squash. All right. Once again, we don't expect to get 16 parts because we're not doing a dihybrid het cross, right? We're not doing a standard F1 cross. So what are we going to get? Mendel works. Big A times little a, 1 to 1. Little b times big B, little b, 1 to 1. Put all those together, pile them all up, and bring them down, and we get some genotypic ratio that looks like that. That's our genotypic ratio, right? Big A, big B, little b is a genotype. We get one of those genotypes for every one of those, for every one of those. All right, but now we have to do a phenotypic ratio. Well, big A is a dominant thing over here, so it doesn't matter what all this is. That's just going to be a white. So we know that these 2 genotypes are going to give us white. This genotype gets allowed to have color, so it's going to be yellow. We can just look up here and see that it's yellow. And then this one's going to be allowed to have color, and the color is going to be green. So what we're going to get out of this are 2 whites, 1 yellow, and 1 green, which is a 1 to 2 to 1 ratio, or 2-1, whatever you want. Okay, it's one of those epistatic ratios. Right? All right, why is it epistatic? Because there's two loci. Even though it looks like a classic old Mendelian 1 to 2 to 1, it's not, because you have to have two loci involved in order for it to work. Again, this is our final phenotypic assortment ratio. Right? Well, that's what's underneath these epistatic problems, right? You're either given a ratio like this, or you're asked about it or something, and you have to figure it out. Okay, but it's just that simple. You know, Mendel does the work of the genotype production, and all you got to know are some of these ratios, and that's fairly straightforward to learn most of those ratios. So let's take it and throw it into a problem with some words, okay? So here's a paragraph. We got some plants. They got a red cyanin synthesized from a colorless precursor. Okay, hydroxyl group is thrown on it, make it purple. Okay, fine. Okay, nice little thing to tell you about there so you understand how you get purple, but you know, that's really not part of the problem that's important to genetics at the moment. What's important to us is that in a cross between two randomly selected purple plants, you get the following. All right, so we made a cross and we get this. What is this? Is it top down or bottom up? Okay. The last couple of problems we just did, they were top down. We give you genotypes and say, what are they going to make? Tell me the progeny. Well, here I've got the progeny. So sure enough, this is a classic A, B, C. We've got C. That's these progeny right here, and we want to know something about A and B. Guess what? You're going to be asked to outline this and do all sorts of things, right? So just diagram it. Might as well just thinking about that. What's he going to ask me? He's going to ask me, what's the mode of inheritance? What's the genotype of all these guys that are C? What's the genotype of A and phenotype of A? What's the genotype and phenotype of B? That's exactly what it's going to be asking you, okay? It never changes in these genetics problems. Right? We may give you a different part of the answer. We may give you A and C and ask you about B, or we may give you A and B and ask you about C, or whatever. Here we're giving you a little bit of information about A and B. They're both purple, and we're giving you C. Okay. Now, there's one thing in this problem that I want to point out. This two randomly selected purple plants. Okay. Now you got to watch that. If you see a problem where it says randomly selected, or they were pulled from different parts of the country, or two different growers had the plants, or whatever animals, and it's epistatic, you have to be a little careful. Okay, most Mendelian problems that we've worked, all the complications that we've had, have pretty much been when they were parental plants, they were homozygous for the most part, etc. Well, it turns out in epistasis that you can have things that aren't exactly homozygous. They're not true breeding, yet they can be the same color. You can have purple plants that are a different genotype, okay, and don't breed true, but are, you know, are purple. So when you see this random, just think. Now, that doesn't mean you shouldn't start with Occam's razor. You know, you want to start with the simplest approach. Simplest approach for genetics problem is to take some classically thought of parentals that are homozygous and figure out, okay, is that going to work? If it doesn't work, go back. And I'm just telling you that because some problems you start out that way and it doesn't answer, right? And you go, oh, what's wrong? I don't know what I'm doing wrong. What you're doing wrong is that you made a choice that just isn't correct. You have to go back and think about it. And that's particularly true with epistasis. It won't be true here, but it's true in things. Okay. Anyway, we got A, B, and C. Now, what am I going to ask you? Well, guess what? What's the mode of inheritance? How many loci are involved? You know, what are the combinations of alleles that you need to produce all these? Basically diagram the problem, okay? So what ratio fits it? Maybe I need to use a ratio. Well, if I thought about it, 3 and 9, that's almost a 1 to 2 to 1, right? 1 to 2 to 1 kind of fits that. So maybe it's just as simple. If that's all I had, I didn't already been told it was epistatic. Maybe I think it was that. Well, how do I test that to see if that fits? Well, this is one of those interesting ones where you say, well, um, 1 plus 2 plus 1 is 4 parts. 31 plus 94 plus 20 plus 43 turns out to be 168 apparently. Okay, so I can do a chi-squared. I've got my observed. I can figure out my expected by using this ratio. Okay, and if we use that ratio, it turns out that we would expect about 1/4 of them to be red, 2/4, 1/2 to be purple, and about 1/4 to be colorless. So they would expect about 42 of those, about 84 of those apparently, and about 42 of those. All right, well, we've got our reserved, we've got our expected, we can do a chi-squared, and if we do, it turns out that it's about 4.09. Well, that's not too bad. Okay, it's not perfect, but it's not too bad. In fact, I think with 2 degrees of freedom, what is it, about 5.89 or something is your table value. that you can look up in the book, well, it kind of fits, all right? But here's what's important. When playing with ratios, particularly when we get these more complicated ratios, chi-squared is goodness of fit test. In other words, there might be something that fits it better. What we mean by better is that the closer this observed number gets to 0, that's nearly a perfect fit, right? 0 would be a perfect fit. The smaller that number gets means that you got a better one. So maybe there's something here that we should be looking into to see if we can find it, right? Well, I don't think it takes too much to say, well, what about just kind of looking at it? What about if we just thought about the fact that if we threw out all that stuff over there and just looked at this, there's a ratio kind of hidden right in the middle of this, isn't there? Okay. Do we have a ratio kind of hidden right in the middle of all this? Yeah, it looks like to me we do. We have a 934 kind of hidden right there. So let's try that and see if it works. Okay. Here's 934. That adds to 16 parts now, not 4. So in this case, about 3/16 ought to be red, 9/16 ought to be purple, and 4/16 ought to be colorless. All right. If I do that, my math says it's about 31.5 should be red, about 94.5 should be purple, and about 42 should be colored. That's a pretty good fit. Certainly it's better fit than this, right? And in fact, to bring this out, the chi-squared observed about 0.03, near perfect fit, okay? Now, unless we ask you to do a chi-squared, we might, okay? And our problem does, then you could do this. Otherwise, we try to make them fairly simple. It's kind of hidden right in there. You didn't really have to do all that work, okay? All right, so now once we know that, what is the mode of inheritance? Well, 934 is an epistatic. 934 is one of the epistatics you have to know what it is, and that's called a single recessive epistatic. Okay. 9 plus 3 plus 4 is 16 parts. So, that's all what's going on there. Where 16 parts come from? It only comes from one place that we have to worry about, 2 loci with 2 alleles each. So what's the minimum number of loci involved? Would be 2. How many alleles do they need? 4, 2 each. Okay, 4 alleles at 2 at each one. All right, and now all we got to do is figure out, well, what's the genetic combination that produced all this, right? What's the genetic combination that makes these? Well, go back to my little problem. I said, when you're doing this, just do a 934 comes from a big A, big B giving us 9. Little a in our case gives us colorless. We've already done that. It could have been little b's. It doesn't matter. Once you pick them, you got them. Okay. So I just used my little 9-3-3-1 chart and figured it out. Okay. That's the genetic combinations. All right. So now the second part of the problem says diagram the purple cross. So in other words, you got to figure out what's going on, right? Well, to give a 934, well, we know that they have a dihybrid het times a dihybrid het. It had to be. So that's my A, that's my B. We already know they're purple. We're going to produce a C down here, okay? But what it's asking you is, it wants to see the genotypes that give me the phenotype, okay? So it's asking you to actually do it. Well, it's pretty simple, okay? All you got to do is you got to do a simple Mendelian cross, right? This stuff right here. You're crossing a heterozygote times a heterozygote at the first locus. That gives me a genotypic 1:2:1. A heterozygote times a heterozygote at the second locus also gives me a 1:2:1. I put all those little things together, and it turns out that I get this problem that we've seen before. This is exactly the way we did it in lecture 2, where we pulled out, said, oh, what are the ratios, etc. There's my 934. Okay, nothing difficult, not a huge problem to work on. All right. Hey, that's really what's underneath epistatic problems. It's Mendel works for you, does all the work. All you got to do is pull it out, and then you have to know a few ratios like 9:3:4, 15:1, 9:7, things like that. Okay. All right. So again, next lecture, lecture 5, we'll complicate this a little more and make a slightly more complicated problem sets for you to work on, but they're going to be just as easy underneath if you understand segregation and assortment. If you know Mendel's Big Six by heart, just know it instantly, and you understand assortment, you can work these problems. You don't, you can't work any of them, okay? You're struggling, so go back and try to do it and learn those if you don't have it already, okay? All right.